in terms of cell therapy and lung cancer particularly, all approaches remain in the context are experimental or remain being evaluated in clinical trials. And I deliberately, rather than trying to present early phase trial after early phase trial, I presented a bit of an overview at BTOG this year and focused really on three main approaches, which include TIL therapy, tumor-infiltrating lymphocyte therapy, TCR, T-cell receptor therapy, and CAR-T cell therapy. And we talked about those, focused most on TIL therapy, in part because that's in its most advanced stages. And that's largely because it's the earliest form of cell therapy for solid tumors, which has been around since the late 80s, originally in melanoma and now finding its way into lung cancer. I presented some early phase data from that, demonstrating sort of safety and possible activity, but very small numbers of patients. Operation is challenging to deliver cell therapy studies, partly because there's lots of regulation around it, as you'd expect if you're using a patient's own tissue to generate a product. You need to be able to demonstrate the pathway of that tissue from the body through to the lab and then back into the body. So for good reason, it's highly regulated, and it's taken a while to try and open these studies in the UK, and mainly they're open at pretty academic centres that have the capacity to run these quite complex studies. But then I suppose that mirrors things like bone marrow transplantation. I think more and more we're more au fait with these sort of studies, and everybody's enthused about them. In terms of tumor-infiltrating lymphocyte therapy, there were some good questions at the end of the session, because this form of therapy involves an operation normally to isolate a piece of tumor or extract a piece of tumor, resect a piece of tumor, and then from it isolate tumor-infiltrating lymphocytes, the TILs, which you then expand up to high numbers and or manipulate further before giving them back to the patient. And there were comments that, look, is this really going to be something that's practical for non-small cell lung cancer patients who tend to be a poorlier group of patients overall? And I thought it's a completely fair question. The things that really encouraged me about TIL therapy and lung cancer is that, number one, and we didn't really touch upon this, is that the side effects or the toxicity that people experience, patients experience, it tends to be pretty much all limited to the inpatient admission when they receive the TILs. So once they're through that, they can be at home in the community, recovering, as it were, but not coming back and forth from the hospital every 3 weeks for another therapy and having to go through, toxicities a la chemotherapy every 3 weeks, for example. So there are advantages in that context. And if they are a vulnerable patient, it means you can watch them closely on the wall in a hospital, which is sort of reassuring until they're fit for discharge. And the other thing is that actually people are thinking about these patient groups and, how can we make these sort of approaches more feasible in different groups of patients with tumors that might arise in different anatomical locations. And for example, one of the cohorts that's open in a TIL trial in lung cancer is evaluating the use of core biopsies. So patients don't need to go and have an operation, a general anesthetic, and be fit enough for that. But actually they come into your interventional radiology department, have a few core biopsies taken, and those are used to generate your cell product. And I think that's a really forward-thinking view. And I hope those kind of interventions will make TIL therapy more accessible for a greater fraction of non-small cell lung cancer patients. But in terms of efficacy overall and finding its niche, that's what's happening at the moment in the context of trials, trying to understand where might TIL therapy find its niche in lung cancer. And there are trials in non-small cell lung cancer patients who are naive to immunotherapeutic approaches, which would be interesting. Those that are refractory to immunotherapeutic approaches. So I think it'd be really interesting to see how all of that pans out. Can we benefit patients who historically might not have benefited from checkpoint inhibition? That would be an interesting question. And seeing how these patients do overall will be really exciting to see how these studies read out. Trying to understand how practical some of these approaches are, particularly TIL therapy for solid tumors, leads us into TCR therapy and CAR-T cell therapy and advantages of those. And if we consider TCR, T-cell receptor therapy first, an obvious advantage there is the starting material is whole blood or peripheral blood. So pretty much everybody's a candidate to undergo apheresis. and isolate T cells from blood. So it's a much simpler process than having a general anesthetic and resecting tumor. And the other nice thing about TCR therapy is that they are directed against targets thus far that aren't bespoke to the patient, as it were, but they're found across different tumor subtypes, like tumor-associated antigens, for example. And I think I mentioned NY-ESO-1 in my presentation as an example of a program that's ongoing. That's useful. That's the idea of being able to have an off-the-shelf product directed against a particular target. But still with TCR therapy, you have to have the right HLA haplotype. And so that's narrowing down your population of patients again. So those that are fit to have apheresis and wait for a product to be generated. That is the other issue, that for all of these studies, you have to wait for manufacturing. And then the second thing is you have to have the right HLA haplotype for TCR therapy. But as I said, I think the advantage of TCR therapy is this idea that it's a sort of off-the-shelf product. CAR-T cell therapy has a couple of advantages that extend beyond TCR therapy. So the first, I think the biggest, is that CAR-T cells are directed against whatever antigen you'd like them to be. And that chimeric antigen receptor, that extracellular receptor, binds to its target antigen in a non-MHC restricted manner. And we touched upon in my presentation the fact that downregulation of HLA or mutations leading to defects in antigen presenting machinery is a well-recognized way by which tumors evade the immune system, and a reason that checkpoint inhibitor therapy may fail or later fail. It's a reason that TIL therapy wouldn't work if a expanded T cell product can't recognize any antigens on the surface of tumor cells. But CAR T cell therapy can potentially overcome that as an issue, because it, as I said, it's non-MHC restricted binding. That's number one. Of course, they're much more complex than TCR therapies in terms of intracellular engineering those to have various co-stimulatory domains to promote the expansion and persistence and activity of those cells. But beyond that, also only turning themselves on in particular environments, for example, having suicide switches so you can turn them off, having fourth generation so-called trucks being able to deliver a payload of cytokines, for example, or within a tumor, and turn it into a more inflammatory tumor in a positive way. So that's really exciting. I think going back to TCR therapies, they're getting more complex. Each new generation is better than the other. And so it'll be interesting to see how that plays out. And those are being explored across multiple different tumor subtypes. With CAR T cell therapy, we commented that at the moment in lung cancer or thoracic malignancies, these have been used most against mesothelioma, mainly directed against mesothelin and FAP as target antigens. And one of the reasons that they've been explored in mesothelioma is that for a significant proportion of patients, mesothelioma remains confined to the thorax, which is unusual for solid tumors as a general principle. Giving local therapies, unless it's going to cause an abscopal or systemic effect, is of limited utility normally. But with mesothelioma, that is a slightly different situation. And so they've been exploring the possibility of intrapleural CAR T cell therapy, so injected into the pleural space. And that's interesting. I think that is a bit more reassuring from a safety perspective. Our biggest issue one of the biggest issues, I should say, with CAR T cell therapy and TCR therapy is trying to find antigens, targets, which are expressed at high levels in the tumor, but where the expression is largely restricted to the tumor. I think it's fair to say, I don't think my hematology colleagues would be upset for me to say that I think they've sort of lucked out to a certain extent with CD19 in the context of CD19-directed CAR T cell therapy. So the CD19 is expressed in BALL and diffuse large B-cell lymphoma, and as such, it's a target for CAR T cell therapy there. But it's also expressed on normal B cells, but there's no major clinical consequence of killing or depleting normal B cells. Some of these patients need immunoglobulin infusions, and the B-cell aplasia that's developed appears to be long-lasting. But thus far, and obviously it's still reasonably early days, these patients haven't run into problem after problem with recurrent infections or similar. There is this idea that possibly CD19 is expressed in the brain and in the pericytes, and that might be responsible in part for immune effector cell associated neurological syndromes. But still, I think generally it's a good target that seems to be largely restricted to lymphoma, leukemia cells, and for which on-target off-tumor side effects are limited. In the solid tumor world, we're struggling, and there's lots of work looking at, evaluating candidate antigens for which we could direct CAR-T cells safely. I think what I said in my presentation was what reassures me is this possibility to engineer CAR-T cells so heavily, almost a limitless possibility, so that they will only turn on, if you like, in a specific scenario, perhaps where they're binding to a given antigen and a separate ligand. So you can make sure that that activity is just restricted to a tumor microenvironment, for example, where they would only bind to those two elements. That kind of thing is exciting. And there's lots of preclinical work, animal models demonstrating the utility of those approaches. And something that I thought was very, very interesting, and I thought about it as a principle, was to forget about or not to worry too much about having a target antigen. But the study which demonstrated, a preclinical study that demonstrated you could use oncolytic viruses to force the expression of CD19, to which we already have CAR-T cell therapies generated by multiple different pharmaceutical companies. So you have a solid tumor, you use an oncolytic virus to force the expression of that target antigen, and then you come in with a CAR-T cell product which has already been developed. And that's pretty exciting, actually, and pretty forward-thinking as well. So antigen discovery, or finding antigens which are going to be mainly tumor-restricted, is going to be a challenge, and abundant amongst the majority, not a minority of patients. So that will be something that's going to be really important to see how that story pans out. I mean, obviously, it's an area of high scientific priority, and lots of work's going on in that area. I guess just to finish on the challenges of CAR-T cells, other issues are, the tumor microenvironment and the complexities of that associated with solid tumors. Not to say that, lymphoma is necessarily the easiest setting, but if you imagine a malignancy like pancreatic cancer, for example, that lays down this desmoplastic reaction, there are clear mechanical barriers for the entry of T cells into those sort of tumors, endogenous T cells, let alone CAR-T cells. So it's because of that a lot of people think that combination approaches are likely going to be a way forward, whether that might be anti-VEGF approaches plus CAR-T cell therapy, whether that might be radiation plus CAR-T cell therapies, and there are case reports demonstrating success when those two modalities have been combined. Beyond delivering your cell therapy itself, generally there's an understanding and an expectation that endogenous or host T cell responses that follow will play an important role in ongoing response and hopefully remission for patients. So understanding the wave of immune response that follows an initial attack, if you like, by your adoptively transferred cells will be important. Is there going to be a role for anti-PD-1 or checkpoint inhibitors at that stage? So we may give checkpoint inhibitors post-TILs, post-CAR-T, post-TCR as a method to help those adoptively transferred cells expand and persist. But actually, when you then get second waves, or you get the induction of endogenous immunity following initial tumor cell destruction from your adoptively transferred cells, are we going to need to come in with immune modulatory drugs at that point too? So as you can tell, lots and lots of unanswered questions, but so much work going on in this space. It really is exciting, and operationally, more and more of these trials coming through. The more we do, the easier it will be to deliver. And so I think the next 5 to 10 years can be really, really exciting in this space.